Football was Teddy Bruschi's life. He woke up every day ready to train to maintain his success as an NFL linebacker. As the Patriots' starting inside linebacker and defensive captain, physical excellence was key to his life. This all changed the morning 3 days after the 2005 Pro Bowl when Bruschi suffered from a mild stroke. A blood clot formed and prevented blood flow to the brain. Without the oxygen that blood carries, brain cells begin to die. Once the clot dissipated, Bruschi was left with partial paralysis and severe vision problems. Imagine the struggle going from one of America's most athletic people to not being able to stand on your own. Sadly, Bruschi was not alone in this struggle. Every year, nearly 800,000 people in the U.S. alone experience a stroke. However, only 35% of stroke victims recover completely or with minor impairments. Even if you are not a pro athlete, many careers require some baseline of physical capability, even if it's just holding a pen or being able to walk from your car to your desk. Developing optimal rehabilitation methods and research on the mechanisms of stroke recovery is crucial in order to help more people return to the lives they love. Physical therapy is an established road to recovery. However, new research points to the biological mechanisms behind memory and mobility recovery through exercise. Today we want to walk through some of these mechanisms in order to show you the importance of exercise in stroke recovery. Scientists at Fudan University in China showed that exercise post-stroke improved motor function in rats. Normally, in order for a person to move around, neurons in the brain play a big game of telephone. Neurons from the outer parts of the brain, called the cortex, relay messages to the middle of the brain, called the striatum, or the inner part of the brain, called the substantia nigra. The striatal neurons can also send their messages to that same area. Then, the messages received by the substantia nigra are passed along to other parts of the brain that control motor function. To study the effects of exercise on stroke recovery, the scientists induced stroke in adult rats. Half of these rats ran on a treadmill post-stroke for 30 minutes per day for 28 days before analysis. Using fluorescent indicators, they were able to observe the formation and growth of new neurons. Significantly more new neurons were observed in the cortex and striatum of the rats that exercised. These neurons were also more likely to connect to the substantia nigra. Therefore, communication pathways were better restored in exercising rats. To show functionality of this pathway, the researchers placed rats of the experimental and control groups on a rotorod, a process similar to a log rolling contest. The rats that exercised post-stroke were consistently able to stay on the rotating rod longer than their non-exercising counterparts. This endurance increase shows that the improved neural connections from exercise correlate with improved motor function. The scientists also demonstrated that the expression levels of growth factors in the brain differed between the two groups of rats, indicating a potential pathway for the observed motor improvement. Brain-derived neurotrophic factor, or BDNF, promotes neuronal growth, and vascular endothelial growth factor, or VEGF, promotes vascular growth. Since more blood vessels allow oxygen and other vital nutrients to be supplied to growing neurons, VEGF and BDNF work together. Rats that exercised post-stroke had high levels of these proteins, which is consistent with multiple previous studies. Additionally, NOGO-A, a protein that is known to downregulate neuronal growth, was decreased in the exercise group. The decrease of NOGO-A supports the neuronal growth and mobility recovery that was observed. Not only does exercise improve motor function post-stroke, but a group of scientists from Taiwan found that exercise can improve spatial memory as well. They induced stroke in rats and set them on a no-exercise, low-intensity, or high-intensity regimen. Low-intensity exercise involved treadmill training for 30 minutes per day for 14 days at a pace of 8 meters per minute, where high-intensity exercising rats ran for 20 meters per minute for the same time period. The rat's hippocampi, the main area of the brain associated with learning and memory, were assessed for protein levels. One protein, synapsin, increases the number of neural connections, called synapses. PSD95 is a postsynaptic density protein that is also positively correlated with the number of synapses. Low-intensity exercising rats had higher synapsin, PSD95, and BDNF levels in their hippocampi compared to both the high-intensity exercising and sedentary rats. These results are exciting as they indicate that exercising can result in more neural connections, which could lead to improved stroke recovery. The scientists also tested to see if these molecular changes correlated with structural changes in the neurons themselves. They measured the complexity and density of the dendrites, or parts of the neuron, that received the signals from other neurons, and found that low-intensity exercise did in fact increase both dendritic complexity and density in the hippocampus. To test if these differences actually resulted in an observable change in memory abilities, the researchers used a kiddie pool filled with water with a hidden block inside. The rats must find the block in order to stop swimming, and the block is placed in the same location each time. Therefore, the rats with the best spatial memory will swim straight to the block area once they have done the trial a couple of times, since rats strongly dislike to swim. Rats, in the low exercise group, again performed the best in this trial, showing that the molecular and neuronal structural changes seen previously correlate with better spatial memory. One possibility for why high exercising rats did not perform as well as low exercising rats could be due to an increase in stress from the amount of exercise they had to do. High exercising rats had higher levels of corticosterone, a stress-related molecule. Additionally, high exercise has been shown to inhibit levels of synaptogenesis in the past. This could explain why excessive or stressful exercise was shown in the study to be similar to doing no exercise at all. In summary, exercising post-stroke is a great way to restore both motor function and spatial memory. Exercise leads to many positive molecular changes that may help to speed up the recovery process and lead to better health after rehabilitation. Teddy Bruschi was fortunate to recover fully from his stroke and was able to return to the game he loved. However, all strokes are different and can result in varying levels of initial mobility, yet research shows that low-intensity exercise is beneficial for both your body and your brain. For more information on types of exercise currently used in stroke rehabilitation, please follow the link in the description. Thanks for watching!